It's my mistake. Thank you. Hi, thank you for staying so late. I changed my title and subject totally because I thought it was not really related to the topic of this conference. And I would like to be entertaining as much as possible because it's the end of the day. So I'll start with two comic reliefs. One is that is something I've written, not now, but in response to the concerning lectures we heard this morning about faked articles, etc. So I think, I don't know how much faked articles you're writing or reading, but you're definitely getting, like all of us, a lot of invitations to write something. So I was trying to develop, rather than artificial intelligence, natural intelligence that would allow me and you too to spot right on an invitation for a faked article. Here it comes. Dear doctor, fill in the blank. You are hereby invited to publish. Our focus is urology and also some plants. Be quick, today nothing is rubbish. Our IF is climbing, it's 2.35. Your paper we promise will be archived. Please also consider to join the editorial. When you are dead, we will publish a special memorial. We were deeply impressed by your recent on genes. Looking forward to hear you. Sincerely, Irene. So if you see something like that has this picture, don't take it. Actually, other than this, I don't need the projector, and I would love to have lights. So can we do that? So we were talking about the promises and dangers of AI, and to me, I'm totally excited about it. AI, ChatGPT, large language models. Can you turn this on?
Try to turn on.
They're exciting, they're surprising. They're even surprised for the good the people who develop them. This doesn't usually happen in technological developments, right? And I think that it opens a realm where we scientists that are used to study things that no one knows, right? Like E. coli or photosynthesis, we may have something to contribute to that. And I'm studying a lot ChatGPT. And one of the things that I'm doing with it, and I'll devote 5 minutes to tell you about that before I go into my biology, is that I'm trying to understand its appreciation of sense of humor. How good is this sense of humor? In fact, one of my desires when I retire is to be very serious about jokes and try to do a classification of all the jokes. I have a feeling that although there are many, many jokes, they come from a much limited types of jokes that share a comic element. And two jokes can share a comic element even if their text or narrative is totally, totally different. So here's an exercise I did with ChatGPT. I told it 4 jokes, and I wanted to see if it can tell them, classify them. Joke number 1. What is number 1? Yeah. Man suspects that his wife is becoming deaf. She's losing her hearing. So he goes to the doctor, asks, what should I do? The doctor says, first you have to diagnose her. And he instructs him how to do that, and he does as the doctor says. So he comes back home in the evening, opens the door, his wife is in the kitchen with her back to him. He opens the door, says, and the doctor says, hi honey, what's for dinner? No answer. Goes half the distance, says, hi honey, what's for dinner? No answer. Goes to her shoulder, says, hi honey, what's for dinner? She turns around and says, for the first time, chicken. That was fun. Second, a 22 years old says, when I was 18, my father didn't know anything. Now I'm saying, wow, he learned so much in the last few years. Third is a real story. When my son was 5, he had a headache for a whole week. And my wife said we should take him to the doctor to see what's happening. So I took him to the doctor. He wasn't really impressed, but he said I can do a blood test for you. So my son, 5 years old, he whispers to my ear. He says, "I don't understand this, doctor. The headache I have for a week, and the blood I had since I was born." So, and third is this, and fourth is this. A man had to go to a shoe repair because his shoes were torn. He goes to the shoe repair, to the shoe person. He gives him a receipt. He said it will be ready tomorrow. He puts the receipt in the pocket and goes away. A day passed, 2 days, a month, 2 months, year, 2 years, 20 years, 40 years. He has kids, grandchildren. One day for Purim, his grandson wears the jacket of his grandfather, and he pulls the receipt from the jacket. He says, "What is this?" He says, "Oh, I forgot about the shoes." So the grandson says, "So let's go and take them." He says, "No, but it's 60 years." No, we have to take them. So they go to the market, and unbelievably, the shop is still there, and the man is still there, a bit older by 60 years. He goes very embarrassed and says, I gave you these shoes, and this is my receipt. Is there anything you can do? He says, "Okay, wait a minute." He goes to the workshop, comes back, and says, "Tomorrow it will be ready." So, when we compare proteins, sometimes if they diverge in sequence beyond detectable homology, we cannot detect that they are homologous. But sometimes if they have the same structure, when we compare the structures, we see that they're actually the same, very, very similar. We say, oh, these two proteins, although the sequence is different, beyond any detectable homology, they are actually homologous because the structure is the same. So I asked ChatGPT, I told him the 4 jokes, and I said, can you arrange them according to comic themes, such that although the text is totally different, they actually have the same structure, the same comic element. Can you figure it out?
Inversion.
Which one?
Inversion.
Inversions. The first and the second are very specific type of inversion. Because in both of them, both the person who thought that his wife is becoming dead, but actually it's him, and the 22 years old who thought that his father loved so much, but it's actually it's him, you think that the fault is in somebody else, but actually it's you. Right? So this is a comic element. And he got it right. So I thought and by the way, for the second 2 jokes, what is the commonality there? The shoes and then my son with the headache. I think and ChatGPT got it right.
Time.
Exactly, time. Not exactly time. You have a long process and you mix it with a short process. And we know that when we model complex dynamic phenomena, we have to worry about separation of timescales. If enzyme kinetics is involved with lizards in the desert, you have to separate timescales because you can't describe the same process over so many different timescales. So ChatGPT got it right. I was quite amazed. I had to try these jokes. I know them in Hebrew, and I had to. It's also a different matter of culture, but I did translate it and got it right. And then I thought if he does this, he could do 2 additional things that we know from biology or from protein structure. One is the inverse protein folding problem. If you know the theme of somebody who has a fault, but he thinks that it's somebody else's fault, then you should be able to identify more jokes, totally different in context, in narrative, in text. Stories very, very different, but they have the same comic elements, so find them in a database. And second, generate new jokes with the same theme, like the inverse protein problem, folding problem, right? This he did very lousily, I have to tell you. He couldn't generate anything that's funny. So he understands the comic element, he can classify jokes together, but he cannot produce.
Yeah. Maybe it's an inversion and you didn't understand.
What's that?
Maybe it's an inversion and you didn't understand the joke.
Yeah, maybe. I can show you, it was lousy. Okay, that was a second comic relief. How much time do you have?
You have 10 more minutes.
10 more
and
minutes.
5 minutes.
That's all I need. So I'm very interested in biology. within biology and evolution. And what drives evolution, as you know, is mutations. And there's a very basic question in research of evolution, genetics, etc., regarding the optimal rate of mutations. What should be the optimal rate of mutation? If it's low, you conserve your genome, but you cannot explore new solutions. If it's high, you explore, but you don't conserve what you have so far. So it's a trade-off between, as called also in cognitive sciences, exploration and exploitation. If you explore, you cannot exploit. Today you came here to explore. You don't exploit on the things in the lab, right? Tomorrow you exploit. You might explore less. And organisms have to face with this dilemma. What should be their mutation rates? High, low, intermediate. How to determine that? Now, there are lots of solutions and a lot of studies on that. And what's common to a lot of them is that they all assume there is a single optimal mutation rate, and all I need to do as a scientist is to find it, and all the organism needs to do is to evolve towards that optimal mutation rate. But once you relieve yourself from the need to have a single mutation rate, you can vary the mutation rate, and then maybe something interesting come up. So in my lab, we study 3 different axes along which mutation rate varies. One is very well known, and it's across environmental conditions. This is actually called stress-induced mutagenesis. You know it, right? When organisms feel bad, they increase the rate of mutation. This appears to be not just an accident of things going bad, but actually a programmed program, if you will. So they have, for instance, a special DNA polymerase that is activated under stress to operate at the high mutation rate, because when nothing works, you should roll the dice. This is one axis. Another axis is along the coordinates of the genome. Who said that every gene must be replicated with the same error rate, same mutation rate? We study a mechanism, I can tell you more about that in another opportunity, that is based on the coupling of transcription and reverse transcription that feeds into the genome a higher rate of mutation due to all sorts of changes, including RNA editing, perhaps, I don't know what else, that happen during transcription and during reverse transcription in order to allow you to effectively increase the rate of mutation of the genes that are needed now, keeping them silent or relatively colder at other conditions, and keeping in that condition the rest of the genes at a lower mutation rate. So you explore where you need exploration, and or where and when, and you don't where and when this is not good. But the third axis is what I want to tell you in a little bit more detail, and that is the axis of individuals in a population. Imagine that all of us are isogenic bacterial population. We have exactly the same gene. And my life is precious to me as much as your life is precious to me. So we are very non-selfish in that respect, and we are genetically identical. Who said, though, that in contemplating an optimal mutation rate, we all have to have exactly the same mutation rate? Maybe we can toss a coin, and the result of this coin toss would tell us what should be the mutation rate that we would operate on. Maybe you would high, would be intermediate, I'd be low, etc., etc. And rather than committing to one value, despite the trade-off that it comes with, maybe we can circumvent that trade-off by dividing. You will be an explorer, I'll be an exploiter, etc., etc. We looked into the literature with this vague notion, and we found that it actually manifested. In E. coli, Johan Paulson showed that there is variability, cell-to-cell variability, even among isogenic cells in their mutation rates. They don't operate at the same mutation rate. How do they do that? How do they toss a coin? They have a protein called ADA, A-D-A. ADA is a DNA repair enzyme. It's present in a very low copy number per cell. Some cells have 0 ADA, some have 1, some have 2, rarely they have more than 2. Not only that, it's a very low copy number protein, it's autoregulatory in a positive fashion, so that if a cell got a little bit more than average, it will have even more. And if a cell got a little less, it will have even less, so it diversifies the population. So now the portfolio is very varied. Different cells gamble on different mutation rate. This is a DNA repair enzyme. If you have a lot of it, your mutation rate effectively goes down. If you have a lot of it, it goes up. And indeed they show that. So we asked ourselves, what is it good for? And is it a bug or a feature? Maybe it's just noise. Is it a bug or a feature? Does it help them to adapt? Does it help them circumvent that trade-off between exploration and exploitation? And perhaps even a very important additional question is this. Suppose that I was a mutator because I had a low copy number of ADA. And Dan was very conservative, had many others, so he conserves, doesn't allow mutations. Compare now my offsprings to Dan's offsprings, or maybe the second generation or the third generation. If this were a genetic property, then my offspring should behave like myself and Dan's like Dan's. If it was the totally randomized trait, each generation you toss the dice again, then my offspring could switch with your, right? If I were a mutator and you were a conservative, maybe in the next generation, my offspring will be conservative and yours will be. And maybe it's somewhere in between, as befits, for example, what we call epigenetic processes that are not random but not genetic either, somewhere in between, so that my offspring will sample from a distribution around my value and Dan's offspring will sample from a distribution around his value, but maybe the distributions overlap, and maybe in another generation they would overlap even more, and maybe after, I don't know, 3 generations, they will not remember the legacy of their grandfathers being either conservative or explorers. So we asked, is it good to have diversity of mutation rate in a population? And if it is good, what makes it better? What makes it better? Genetic inheritance of this trait, as we usually refer to mutators. Everyone who ever worked with bacteria knows that mutators are thought to be strains that have a mutation that renders them mutators. So that it's a genetic property. Is it a totally randomized process? Or should it be a randomized process? Or should it be a process that is memorized with some fading memory, as suggested by this autoregulation of the other protein that we see in biology? So how to study something like this? It's very complicated. For a lot of time, and when I say we in this project, I mean my PhD student, Gabriela Lubinská, and this was all in collaboration with Yoav Ram from Tel Aviv University, who was Gabriela's second PhD advisor. So for a while, Gabriela and I were thinking, can we inactivate this memory property of E. coli to get cells that either memorize perfectly or memorize totally imperfectly, as opposed to their intermediate level of epigenetic, and it was very hard to do, so we resorted to a mathematical model. In our model, there is a fitness landscape, a mathematical description, a mapping of the genotype to phenotype. Okay? This could be my phenotype, and when I do a mutation, that could be my new phenotype, or genotype, sorry. And the fitness is how good am I, and it could be that the network landscape is very rugged, so I can do one mutation and fall off a cliff, or it could be smooth, etc., etc. There could be valleys, there could be plateaus, there could be all sorts of things. And we experimented in this model with all sorts of fitness landscapes, with various levels of ruggedness. So now we have a population, mathematical population, of creatures that have their initial genotype. Some of them are explorers, have a high mutation rate, some have low mutation rate. And the tunable parameter that we want to see the optimization of which is the adequacy of inheritance of this trait of being a mutator or being a conservative. Again, at one extreme, it's a genetic property, so you inherit it as is. On the other extreme, there's no memory. The next generation forgets totally. And in between, there is the twilight zone of the epigenetic inheritance. You inherit it with inaccuracy. So we let our organisms explore the fitness landscape, and solving it means to go to the to the highest tip, crossing fitness valleys, which is very hard to do because it's very tempting to stay at the local optimum. We know it from other fields as well. Turns out that the solution that maximizes adaptability and that helps organisms most robustly get to the highest point is this epigenetic inheritance mechanism in which they inherit the mutation rate. There is variation in mutation rate, but it's inherited semi-accurately, epigenetically, not here and not there, but somewhere in between, very similar to what we see in biology. So we said, can we explain that? Can we understand why it happens? And the intuition is very hard to wrap your heads around. But, and I can tell you more about that perhaps if you ask me questions. But what I want to say in the last minute or so is that, and this is a connection to the world of AI, etc., is that I believe that maybe there is I don't believe, I speculate, that maybe there is an opportunity here to have yet another lesson that comes from biology that goes into computer science. We know that neural networks are a good example for that. We know that genetic algorithms are a good example of that. And here is a potential new one. Think about a classical problem in AI, machine learning, all that stuff. You have a network, neural network in fact, and you have to optimize all the weights. And again, it's a very rugged fitness landscape, or not fitness, how should I say, utility function, right? Because maybe you change one arrow and then you destroy, you need to change a few in parallel, so there's valleys to be crossed, etc., etc. And what we often do in optimization algorithms is, again, another metaphor from the natural sciences, this time for physics. We do something called simulated annealing. In simulated annealing, you have a metaphorical temperature, and the temperature declines during the simulation time or during the optimization process of the algorithm. The temperature can dictate two things. One is how large are random steps that you do towards optimization. If the temperature is very, very high, you are ready to flirt with very dramatic changes, like high mutation rate. If the temperature is very low, you're very, very conservative and you don't change much. Also, the temperature governs your tolerance to bad changes. If the temperature is high, you're willing to even accept a change that is bad, hoping that at some point it will pay off. And if the temperature is low, you're not accepting bad changes at all, only good changes are being accepted. Simulated annealing is a very well-known concept in optimization algorithms, applied also to machine learning and that stuff, in which you lower a metaphorical temperature with time at a certain schedule, so that at the beginning you're exploring and at the end you're very conservative. We asked, did we just discover simulated annealing, or maybe they are doing the bacteria something better than that? So we let the simulation of our process run twice. Once is that it needs to solve a rugged fitness landscape, and indeed we see that at the beginning they do more mutations as if the temperature was higher, and then they gradually convert into less and less explore and more conservative ones, lower mutation rate. But then in a second round, we took that schedule of apparent simulated annealing that appeared, and we imposed it how it did on the other population. And we saw that it didn't do as well as this population. So although simulated annealing, gradual decline in the temperature, is naturally observed in our system, we saw that they are doing something better than that. And the something better than that they are doing is hard to explain. Maybe I can take, maybe in the expense of the time of questions. But the short of it is that if you're trying to go up and down and get to a very high mountain, if you're stuck with your mutation rate as a genetic property, even when you solved it, you would still have a high acceleration or a high momentum because you have this property of being with high mutation rate, so you slide from the other side. If the inheritance is totally random, you have a high mutation rate, you get some mutation, but you are losing that property in the next generation. Apparently, what biology teaches us is how to couple the mutation rate with number of background mutations that have accumulated so as to slide and stop the sliding process when the problem has been solved. And what we are trying to examine now is whether we can use that as a general optimization principle that could be applied also in optimization of problems in a totally different regime, such as in machine learning, etc. So we'll stop here.
Similar to Monte Carlo, and you change the step size?
Say it again?
You change the step size?
Yeah, so in Monte Carlo, you change the step size, and you usually start with high step size, and you gradually cool the system and go to a low step size. What we suggest that we saw in biology is that the step size is coupled for each solver to its own success so far. It's not that we impose from the beginning one schedule of reduction of step size, irrespective of the success of each solver, but rather they get the injection of the high step size when they need, and they don't have to commit for it for too long, but for long enough in order to allow them to cross some fitness
So
values.
it's basically when you, it's just reminds me. So you can basically follow how many steps you have accepted. Based on the acceptance rate, you can increase or decrease the step size. That would be some type of memory that you're talking about.
You can do that. It might be that this is what I'm talking about. The only thing that really happens in our simulations that appears to be doing better than a simulated annealing of predetermined rate of decrease in step size is that you have a population, they need to accumulate mutations towards solving it. They go in and out of the stage of being mutators at a certain rate, and if that rate is tuned to be optimal, then they appear to stay with it, but only to the extent that they need it. Not less and not more. Less would be if it were random, more would be if it were genetic, and the epigenetic mode of inheritance appears to somehow optimize the connection or the coupling between the amount of mutations that they have gathered so far and the extent of genetic acceleration that they still have. So this is the premise, I think, of this notion.
Yes, Ute.
Yeah. I have actually a couple of questions, but I ask only two. The first is it possible to have isogenic organisms with different amounts of what is ADA, right? ADA enzyme. After all, it's an enzyme. It's coded for by genes, and if the amount is different,
Very good question.
should they be isogenic? I have another question. No, you can talk.
Can I answer that first?
Yes, sure.
Yes, it's a very good question. Is it possible for cells that are identical in their genome to have a different copy number of an mRNA for a gene or a protein from that gene? And the answer is that yes, very much so. This phenomenon is called noise in gene expression. And we call it noise exactly because of the variability that it exposes. And this notion of phenotypic diversity amidst genotypic constancy or identity is a very strong notion in biology. It's very surprising. At a second thought, it would have been surprising if it weren't the case. Because although me and you may have the same genes, at the end what executes all the regulatory programs in the cell are random processes of collisions between transcription factors and DNA, and ribosome binds an RNA and produces a protein. So you have the book, but at the end the cell does with it whatever it wants. This is what we call in Hebrew, you have a genome, but the molecules bump into each other and they don't read the genome.
But the randomness, as Stas Schwarzman has pointed out several times, the randomness of these collisions is in the end limited by the genome. I mean, it is not an unlimited randomness. But
absolutely. This is why I said, of course, everything is expected and the permission is still given. It appears that even the extent of randomness is genetically encoded.
Yeah, it is. Yeah. And no, the other question is, I think you are talking about bacteria, right? When you talk about these epigenetic things, and I wonder what the epigenetic interactions are, because I know in eukaryotes the epigenetics factors and chromatin modeling, they are basically also coded for by the genome, by genome factors or by the enzymes which put the markers there. And I don't know what you were talking about in bacteria. And isn't it possible, instead of epigenetic interactions, that it is kind of a horizontal gene transfer of plasmids or something?
Yeah. Okay, it's another good question, and I'll say this again. I'll be a little bit philosophical, but it will suit you. When we say epigenetics, it might be that there is variability in the meaning of this word in this room and among any set of biologists. For some people, epigenetics are the markings that code the DNA, methylation, histamines, etc. And then, indeed, most of them are unique to eukaryotes. In fact, not all eukaryotes have all of them. But for me, epigenetics is everything that is heritable beyond a physiological memory. And then a lot of chemistries in the cell might feature epigenetics even though they are not the DNA chemistry. For example, some proteins that act as prions can template themselves and replicate a conformational change, conformational information that one of them has. And Susan Lindquist, who Leslie did her postdoc with her, unfortunately she passed away, had this brilliant idea that epigenetic inheritance by prions could serve as a way to conduct information non-genetically for a while until it's being forgotten. Oded Rechavi from Tel Aviv University has shown that RNA can serve as a chemistry for epigenetics because it can replicate itself DNA independently.
Yeah, but
so
it has information. It is not.
It is information.
It's not,
always
it's
information.
not epigenetic.
To me it is, but we don't
Okay.
have to argue the terms.
Okay. Thank you.
Yeah. I'm suggesting to broaden the scope of what could be considered epigenetic.
No, I have also a broad scope.
Such beautiful thoughts, in fact. I wonder whether the model that you talked about refers only to organisms that are unicellular, and in that way the organism is the reproducing unit. So how do you envision that this model would apply to metazoans, to multicellular organisms in which only part of their cells are the reproducing unit, and whether there are mutation rates which differ between different individuals, different groups of individuals? How does this work in your eyes?
Very good question. I haven't thought enough about it. Well, one easy answer, but I'll try to do the harder too. One easy answer is to think about cancer, which is actually a microorganism within a multicellular species. So a potential prediction is that maybe cancer may also manifest this epigenetically inherited mutation rate. Cancer is known to have variable mutation rate. And the modes of inheritance upon mitotic cell divisions, not germline. Mitotic cell divisions could be complex, and if this is an evolvability means what I just described, namely a tools with which you can solve complex fitness landscapes, maybe cancer has thought about that. But you asked a tougher question, on which I haven't thought, to be honest. I think that if the principle that we described here were manifested, for example, in sperm cells or in oocytes, that would have amounted to them having different mutation rates, very hard to measure, and some process of semi-accurate inheritance of a mutation rate during, for instance, spermatogenesis. But I don't know how to measure it. I don't know if the logic applies. I don't know. I need to think about it.
In the aging.
Yeah.
Yes. You spoke about the differences in the rate of evolution, but applying similar logic, one can think about maybe different level of mutation rate in different on the subset of the genome. So if you have genes that you believe reach to optimum, you want less mutation there. So what about alteration in the mutation rate in the genome?
Yes. So if you remember, when I started, I said the question is what's the optimal mutation rate? There was a trade-off. Who says that there needs to be a single mutation rate? Maybe it could vary. So we examined three axes along which it could vary. One is that I talked about, varying along individuals genetically identical, but individuals in the population. Second is between conditions. If it's hot, then maybe mutation rate would be higher. The third is exactly what you're saying, along the coordinate of the genome. And we hypothesize that this mechanism of coupling transcription and reverse transcription, genes going in cycles of transcription and reverse transcription, using RT services that they migrate from all sorts of reverse transcriptases that occur within our cells. The alu elements owe their life to the lines RTs. And in yeast, for example, as you know, I think there's another such element. And we suggest, and we are doing experiments to test the notion that indeed, when a gene is expressed, a bit similar to transcription-coupled mutagenesis, when a gene is expressed, if it goes back into the genome, then it harnesses RNA transcription mistakes, perhaps editing events, modifications, splicing, copy number information, etc., to enrich or to inform the genome. And when it's not expressed, it's not. So it's not exactly what you want. That the mutation rate will depend on how good is that gene. I don't know if the cell can know that, but it definitely knows that it uses that gene now. So maybe keep a low, cold kind of metaphorical low temperature for the rest of the genes, and high for this gene, but only in the condition in which it's expressed. We're still examining this.
Depending on tissue specificity versus.
Yeah, although we do that in yeast, so no tissues.
Okay, so one last comment. I would define it as an integrated strategy. I wouldn't use the term epigenetics because you may have plasticity, yes, and on the other hand, back edging or coil flipping. So it's an integrated strategy because you can do the fine-tuning using plasticity. I think this is the best definition. But thank you, Tzachi. And all the speakers, Uri, Shai, Tzachi, and all the speakers that we had today, I enjoyed the symposium very much. I learned a lot. And I'm looking forward for the next symposium.